All right, I'm gonna go ahead and get started. So I'm Andrea Papaleo. I'm a software engineer here at Broad Institute of Harvard and MIT. I'm part of the imaging platform. Um, and I'm gonna talk to you about Piximi, which is an image classification and segmentation tool in the browser. So yeah, the reason why we're trying to expand upon the tools available to us for deep learning is because it gets us from data from the bioimages to actual results and insights in a very quick and efficient way. I'm sure everybody kind of knows that. So instead of taking images with these microscopes and getting the cells and having to annotate them and classify them, it's much easier to do that using deep learning methods. One of the biggest pain points with that is understanding and learning deep learning and machine learning, especially as someone who is primarily a biologist who spent their career, their schooling, learning biology. They don't want to try to learn a whole other field. There are plenty of tools out there that you can use that can do this for you on your computer. So there's Cell Profiler, which is a very popular tool. The problem with a lot of the local tools is that there's pain points when it comes to installation, making sure that you have correct versions of Python, correct versions of Java. And so a lot of times researchers will try to use that, start downloading it, start trying to install it, and find that that whole process of installation is more trouble than it's worth. And they think that using the tool itself is going to be as hard as it is to install it. So what we want to try to do is alleviate that. Right. So we want to be able to leverage the power of deep learning in a way that's equitable and universal. So we want to put this vastly powerful way of classifying and segmenting these images into the hands of any biologist, regardless of their computational experience. One way to do that is by putting the tool on the web, right? So with that, there's no installation. You go to the website. In this case, you go to piximi.app, and it's there. It works. You can upload your images. You can, run these classification tasks, segmentations, and you don't have to worry about having the, correct tools already on your computer. You don't have to worry about any of that installation. And especially now with the browser becoming more powerful, it's a lot easier to do that, especially when it comes to accessing the GPU on your own computer, which is another benefit specifically of Piximi, is that all of the work that you do, the images that you upload, all happens locally on your computer. So you don't have to worry about what's going on with your data when you send it off to a server somewhere that does these computations, that does any kind of inference. It's on your computer, private, and it's all right there and easy for you to do. Right. So the couple of issues is that there's not much library support in JavaScript as there is in Java and Python. And you would need internet access to use this tool. But unless you have a Docker to run it locally, or in the future, we would like to take Piximi and make it so that you can run it locally and help to push JavaScript and work on these libraries to expand what you can possibly do in the browser as well. So again, with Piximi, you have your images to discovery. So your users who care about whole images, care about classification tasks, and they want to be able to measure or take measurements of their images as far as average intensities. They want to see their histogram, number of objects in the image, and then people care about objects, which is segmentation and annotations. So Piximi is trying to be able to encompass all of that and really provide everything that you would need. Well, not everything, but everything that you would need to start your analysis of your research there. So I can just save the slides, just kind of go ahead and show Piximi in the browser. So can everybody still see the screen?
Yes, we can.
Okay, great. So this is what happens if you have your computers. You can also go to piximi.app. This is what you're faced with when you first load in. This is what we call the experiment view. Right, it comes with this default image here. You can upload your own, pretty much your own files. If you have a Piximi project already, you can upload those projects. You can upload any kind of image that you have, or you can use any of the example images or example projects. So I'll get into an example project in a little bit, but first I'll just kind of go around and show what's possible here. So you load up your images here. You have categories. You can create a category. Right, so just new category. You should, as a recent push, right? Recategorize your images. And then it's simple here to go, and if you want to make any changes to that image, you go to the annotator. So right now, this image comes pre-annotated, but you can delete those. And then we have a suite of tools here that you can use to create these annotations. So again, just regular rectangular tool, ellipse tool. So your basic shapes, polygon, pen, lasso. And then we also have these thresholding tools. So you click, right, select an area, and then zoom in and then confirm. I think there might be, should be able to change the thresholding, but that is not here. That's fine. Right. And then you can go through again, select on an image, and then you can yeah, sorry. So, some pain points here, but you can recategorize them. And yeah, so you go back, you can save your annotations. And they show up here. So if I just refresh and start off with the initial image and the initial annotations, you come here, and then all of the pre-annotation or the pre-segmented image and the annotations created by that show up. So yeah, let me get into an example project here. So we go with this human, we draw a cell cytoplasm crops. So you'd go through, you'd load this image. It's deserializing now. It takes a little while for that to happen. Right, so another thing you can do is recategorize the way these show up. So you categorize them by category. So now it's showing you all of the annotations. These are all the cell membranes. You can hover over them, get some information about these segmentations here. Go down, you have your nuclei. Right, and then there's no unknown, okay. So now, again, this project has no annotations, which is why you saw all the annotations go away. This new project is loaded up here. And again, in this image viewer, or this image-based view of the experiment viewer, It shows you the category, some details about the image, number of channels, planes, what partition it's part of. And same thing here, you can organize it by category, random, what have you. So now these are, go back to classifier, categorized by their stain, so cytoplasmic, GFP, nuclear GFP, but the colors aren't necessarily what you would expect them to be. So we want to change that, right? So that's why. So we can go here, select all the images, switch the annotator. And then within here, we can adjust the colors of the image too. So you go to channels. And so basically, because we don't know necessarily how you want to map your colors to each channel, the default is just RGB, but that's fine. We can come here, channel 1, we can change that to green, channel 2, we can change that to blue. And then we can adjust the maximum minimums here, right? So if we have something, essentially what we want it to look like overall, we can go ahead and apply it to all the images open in the annotator. And we opened all of the images here. So it's going to apply the color changes we made to all of the images in the experiment. Basically what it's doing is going through and updating all the tensors. In the background, we use TensorFlow.js to do a lot of the deep learning tasks. So, right, it's going through, updating the tensors, generating the sources for the images, and then you'll see once that's finished, those changes will be updated here. Right, so let me change. Now we'll go back. We want to save the changes that we just made. And now we're back in the annotator view. And let's just go back and organize by category again. So now we pretty much have what we would expect to see for the staining. So we want to try to classify it, right? Because we have a handful of unknown images or unknown categories for these images. So we can go here, we go to fit model. And so within here, we have a couple different options. So there's pre-processing settings, so we can change the crop type, number of crops. We can rescale the pixel intensities. The architecture settings for the classifier, we can use either a simple CNN or we can use MobileNet, but we'll stay with simple CNN. The inputs, the rows and columns, number of channels, it's all customizable. The optimization algorithm, learning rate, batch size, number of epochs you want to do when you train. You can change, and then the dataset settings. So you can change the training percentage, so how much you want that split between training and validation, whether or not you want to shuffle it on split. You can change all of those, and we try to expose as many settings and different options as possible without, exposing everything. Because if we would expose everything, if you can change and fine tune every little part of the model, then it's no longer accessible for beginners. It now takes someone who's more well-versed in deep learning to use the tool. And that's not necessarily the demographic that we're looking for. So once you make your changes here, you would just hit fit classifier. Goes through. As it's training, you can see some real-time information here. You look at accuracy per epoch. And then it's done. You leave here. Now you can evaluate it and see how well it did. So we get a confusion matrix and then some other metrics here you can look at. So we can see for some of them it did pretty well. So the cytoplasmic GFP, no GFP. And so for anybody that doesn't understand, so this is the ground truth versus the prediction. And so pretty much it's saying that it predicted all of the cytoplasmic GFPs, no GFP, well, all of them in the training and validation sets. So once we kind of were okay with that, we can go ahead and predict. So now this is going to apply those changes in those categories to all of the unknown images. So if you want to just view that, we go and hide the labeled images. Go here, order by category. So now we can go through and try to see, how well it did. So if we're looking at cytoplasmic GFP, we're seeing that like, okay. Well, one more thing to mention is that one way that you would tell what's predicted and what's labeled is by this little icon here. The labeled images is just this tag, solid on the left or a straight line on the left and pointing on the right. We hide those. Now these predicted are pointing on the left and right. So we can go through and look at these, and so far so good, not too bad. And then we say, oh, this doesn't necessarily seem right. So full disclosure here, I am not a trained biologist. So I don't, I haven't spent a lot of time looking at the images, and I can't really tell like from first look exactly what should or shouldn't be no GFP or nuclear GFP. But these do kind of look, a little greenish in the nucleus. If we go through and try to look for some more, this definitely is. And so, yeah, so we know these are wrong. So we can go, we click on that, and then we can recategorize that nuclear GFP. And then we can keep looking and say, okay, this is blue. It's pretty blue. And then recategorize those, no GFP. So we finished, we went through, we made the kind of choices. We don't want to go and try to pick all of them because we're trying to train this model for further use. And if we just kind of picked everything itself, it wouldn't be able to predict on more unknown images. So we did our cytoplasmic GFP, we can hide that. And so now we're looking at no GFP. And again, this, might be cytoplasmic. I'm not sure, but let's click on that one. Maybe there. Let's look and see if there's any more like egregious ones here. Okay, that's very much cytoplasmic. All right, so we can go through, recategorize those as cytoplasmic. And then once we're done with that, we can look at nuclear. And so we can see, maybe that's cytoplasmic. That's not too bad. So we'll go ahead and leave that. And then we'll look for, that looks like no staining or no of the proteins. And we'll say that's none. So once we can do that, we can clear the other predictions, right? Go back and let's go ahead and show all of them again. We can continue the fit. And again, look at our model, see if it does any better or not. And it more or less looks about the same. And so you can tweak the settings, change the batch size, go through, and it's an iterative process. But when you're all said and done, you can go through, you can save this project, and it's going to save the model and the other information there. So I'm not going to go through that, but just going to do a reload, and then I'll go ahead and real fast go through the segmentation since I only have a little more time. So, right, just delete all the annotations, just start with a fresh image here, save those changes. Then we can go to segmenter. Currently, we don't have a trainable segmenter, but we do provide a couple pre-trained models. So we provide, you can use a Coco SSD, which is more just a generic segmentation model. Or we have Stardust, VHE. Both of these run locally in your browser. Currently, CellPose is a little more tricky, so that we have to run elsewhere. So that's run on BioEngine. I don't know if anybody's familiar. but we can go ahead and select that, open the segmentation model, and then go through and predict. So it's going to take some time. It's, again, it's remote, so it's going to take time to go off to BioEngine, do the processing, and then send the information back. All right, so it did that. So now it came back with this new nucleus category, go to annotations, and it's looking fairly good. We can select one of those, go to the annotator, and it's already pre-selected here at the annotator. And we can look through, we can see how well it did. And if it's great, go ahead, deselect it, look at other ones. So again, this is maybe not necessarily the image that you would use for cell pose, because obviously these segmentations that are created are fairly large, and it's capturing more than just the nucleus. But this is just an example of what can be done here. We'll just save those changes. And then, yeah, I think I have a couple more minutes. If anybody has any questions, feel free to ask. Let's see. 2 in the chat. So yeah, what's the limit of the computer power of the uploaded image? I'm not quite sure what you mean by that. The limit of the computing power is essentially what your computer can handle, because it does run locally. If you have extremely large images and very many of them, it's not going to run as quickly on your local MacBook or whatever computer you're using, if you don't have the processing power. Eventually, we would like to be able to build it out so that you could send off these images. You can specify your own backend to do these calculations on. But currently, it's limited by whatever computer that you're currently using.
What kinds of computers can you use it on that note?
Right. So right now we tested on Intel and M1 MacBooks. M1 MacBooks have some issues here and there. You have to kind of adjust the some of the sizes. So if we go back to the classifier for this image, architecture setting, you might have to decrease the number of input rows and columns as you're doing the classification. Otherwise, it'll have issues doing those kind of tasks. But other than that, it also works on your phone. It'll work on an iPad. You also have touch support for the annotator, so you can use like a Apple Pen or any other stylus to create these annotations. So it'll run on low power devices, your computer. You can run on desktop. Yeah. So if you want to do your segmentation and classification on the go, you can. We are also thinking about support for different image formats. So that's the .heic. So if you can take a photo with your iPhone or whatever phone you're using, and then upload that into Piximi, and then do some classification tasks or segmentation tasks with the photo that you upload as well. Yeah, you can also change light mode, dark mode, sound effects. As you're going through, you have this nice help menu that can help you, try to do what you're trying to do. right? Saving projects, creating categories, opening images. Switch over to the annotator. This tells you manipulating the canvas, creating categories, new annotations, changing existing ones. So you have this nice little help menu there. Yeah. And then you can also export these annotations in different kind of file formats. So, Piximi formatted JSON, you can do labeled instant masks, binary instant masks. So if you want to make your annotations in Piximi and then export them and use them in some other tool down the line, you can also do that. To extend the interfacing to BioEngine. Yeah, currently we would like to be able to work and be able to use any model that's currently on and being hosted, like and running on BioEngine, providing that it has the pre and post processing data there. One challenge with that is understanding and knowing what the structure of the output's going to look like in order for us to be able to display the annotations and work with the annotations. So for instance, Stardust uses a specific way they have these annotations by having, I think, like a 33 or a length 33 array where the first index is the center, or the probably that's the center, and the rest are lengths to the edges around and angles. Yeah, so we can do classification. So real fast, it's going to work on n channels. So if it loads quick enough, right, example project. So like, for example, you have this MNIST, right? It's just grayscale. Right, so, oops, hold on a second. So yeah, so MNIST, we can go here and quickly run this classifier. Go ahead and predict. And then it goes through and it makes the predictions. So again, go here, hide labeled images. We can see how well it does. So pretty well, except 8, 2, 8, couple, yeah. So these things are ones. And again, you would go through, you would kind of recategorize the predictions, retrain the model, and just keep improving it like that. So segment one channel and a multi-channel image. So currently, there's no way to separate these annotations by channel. I mean, you can go through, you can turn off these channels and create the annotations based on what you see here, but they're still going to be, if I'm understanding the question correctly, they're still going to be on this image. But if you have like a Z-stack, then you can annotate the different planes of the image on different Z-stacks, and those annotations will be on those specific planes. But as for channels, like it's not, the annotations aren't specific to like channel 1, channel 2, or channel 3. They're just specific to the image and the plane. Oops. You're welcome. So I think we're at time. So if there's no other questions, I will thank you all for coming.